Hi everybody, thank you for coming along to our webinar today, another one of our many webinars this year. Today's webinar is entitled Correlative Submicron IR and X-ray Fluorescence for Molecular and Elemental Imaging of Neurons. And today I'll be joined by Professor Oxana Klementieva from the Neurology Department and Head of the Medical Microspectroscopy Group from Lund University in Sweden. So just before we start, a couple of quick, housekeeping rules. Throughout this webinar, you can type in any questions, comments maybe, in our chat box, and we'll endeavour to answer as much of those as possible by the end of the webinar. Anything that we can't, of course, we'll get back to you via email ASAP. And this is a recorded webinar, so this will also be available for on-demand viewing later on. And really with that, I'm going to get straight into it. The outline today, in fact, I'll be doing the first half of this, I should have said. I'll go through, and I really want to introduce and remind or make aware some of the current limitations when it comes to infrared and Raman microscopy. I'll take you through how O-PTIR overcomes those limitations, how it works, and how it overcomes those limitations. Then I'll take you through a range of applications, mostly bio, but also a few non-bio applications to give you a sense of the broad application base that we appeal to. And then in the second half, I'll hand over to Oxana for the main part of today's webinar. So as I said, the first part, I'll take you through some limitations when it comes to infrared. I call these accepted and known limitations that are really just because the technology is at its fundamental limits. And as far as infrared spectral microscopy is concerned, probably first and foremost and primarily limited infrared spatial resolution. That's just because we're working with long wavelengths. You're limited to in the order of 10, maybe even sometimes 20 microns. Often also the best quality spectra is obtained in transmission mode. So again, I'm talking here in terms of traditional IR. So this requires thin cuts, which might be fine for some things, but for many samples, cutting them to 5 to 20 microns or thereabouts can be difficult to impossible. Some of that can be mitigated with the use of an ATR tip as an accessory, but this requires intimate contact. Precise targeting can be difficult. There's risk of cross-contamination. You can damage the crystal. You can damage the sample. Speaking of which, in fact, here's a picture of exactly what happens when the sample is damaged by the ATR tip. Moving on, there's also the issue here of liquid nitrogen cooling required for these cooling detectors that are used typically in infrared microscopes. These detectors have hold times that are in the order of 6 to 8 microns. 6 to 8 hours, I'm sorry. And if you're doing any long or overnight measurements, this may well require some late night visits into the lab to top up your liquid N2. And if there are any people, I suspect there are a few people here on the call who know the pain of going to the lab at 2 a.m. to top up the liquid nitrogen. But I'm going to leave the best or perhaps the worst to last. And that's really the issue here of dispersive or scatter artifacts. As I said before, the best quality spectra obtained in transmission mode. So imagine that's a thin film of PMMA, and we measure that in transmission mode. The spectra look great, nice flat baseline, nice symmetric peaks. But if you take the exact same material and make it into a bead of, say, about 15 microns, well, the spectra don't look so good anymore. You've got some weird baselines, it's offset. Worse still, your nice carbonyl peak is now split into two, yet it's the same material. But you make it a 10 micron bead or a 5 micron bead, the spectra change yet again. So it goes to show that the spectra are rather particle shape, size, and surface topology dependent. And that can be a problem if you're wanting to do some interpretation of the spectra or to conduct some library searching of the spectra. You don't want the particle shape or size or surface roughness to confound your interpretation or your library matching. On the Raman side, first and foremost, it's got to be autofluorescence. Many samples, especially biologicals, can have significant autofluorescence, and this often swamps the signal. You can move to longer wavelengths, wavelength excitation, and that has some mitigation effect, but the 1 lambda to the 4 issue results in a significant sacrifice in sensitivity. So it's a painful trade-off. And that sensitivity issue is even made worse by the fact that you're starting off on a low base. The Raman cross-section is sort of 10 to the 8, a lot lower than that for Raman. So you're already behind the 8 ball there. And with Raman, you've got to collect, if you want images in Raman, you have to collect hyperspectrally. So you've got to collect the full spectrum at each point in a mapping fashion, one by one, in order to do mapping. And that can be very, very slow. So they can't do single frequency imaging. And that's something I'll touch on later. There's the issue, of course, of laser power sensitivity, because it's a fundamentally limited signal and noise limited technique. You generally want to be pushing the power as high as possible just to get as much sensitivity out of it. But power in the orders of several or tens of milliwatts, which can be required for good sensitivity, are more often than not toxic to any living organisms. Live cell measurements, any solid samples may well damage and burn as well. And when you lower the laser power, that lowers proportionately the Raman sensitivity. And last but not least, we have the issue here of Raman spectra that are dependent on the excitation wavelength. So the spectra can vary, especially if there are visible resonances. I've done some measurements on blood cells, and the spectra are dominated by heme. Not so much the other macromolecules, but it's the heme that jumps out because, of course, it has a resonance with the 532 excitation. But if that was measured at 785, the spectra would look different. There's also the issue of the substrate. So not just the sample, the substrate itself can also be excitation wavelength dependent. For example, glass is a poor choice for 785, but it's okay at 532. And kind of this is a nice segue into the technique of optical photothermal infrared, because it basically overcomes all of those issues I've just listed there. The technique can be described as a pump probe optical spectroscopy technique, where the pump is a infrared laser, typically a QCL, and the probe is a short wavelength visible or near infrared laser. We deliver sub-micron infrared spatial resolution. In fact, it's very much Raman-like in that sense, but you get the richness of the infrared spectra that come out of it. Though we operate primarily in reflection mode, but there's also some transmission, which I'll touch on later. But it doesn't matter whether you're in reflection mode or transmission mode with respect to the probe beam. The spectra that we collect are FTIR transmission-like. So it's as if you'd be comparing to transmission FTIR libraries. There are none of the typical distortions, artifacts, or interference fringes that you would expect in a typical infrared reflection measurement. It's non-contact, far field, so that preserves and maintains the sample integrity and preserves molecular orientation. And the spatial resolution is infrared wavelength independent. That's an important point as well. How might this technique work? I've got a little video to take you through it. So it all starts with the microscope objective. It's a reflective Cassegrain style. Through that, we shine a pulsed infrared laser beam, and being infrared, it's fairly broad, maybe around 10 microns. At the same time, we're shining in our probe beam, the green, it's about 500 nanometers. The infrared generates thermal expansion as it's expanding, and that thermal expansion changes the way the green light is reflected or scattered back. Right, so as we're tuning our infrared laser from one wavelength to the other, we monitor the intensity of the green light reflected back. And it's from that we calculate out what is essentially a pure infrared spectrum collected in reflection mode. So in this slide here, I'm going to compare traditional IR and Raman a bit more systematically, sort of side by side against some of the key attributes for these sorts of instruments. So spatial resolution, if you're using a microscope, you typically care about spatial resolution. And if you want the absolute best spatial resolution, well, today at the moment, you're probably considering Raman and not so much traditional IR. So I've given Raman a green and traditional IR a red because it's worse. If you were worried about fluorescence, it's going to be the other way around. I've given Raman a red and IR a green. If spectral sensitivity is a concern, typically speaking, IR is better, and therefore the speed of measurement is better. Spectral interpretability, or the availability and the extensiveness of libraries, it's going to give that green to the IR because there are about 10 times more commercial libraries or spectra for the IR compared to the Raman. But if you want to work in reflection mode, well, that's how Raman works, and IR doesn't work so well in reflection mode. Water vapor, again, issues in IR, not an issue in Raman. Water as a solvent for live cell measurements, for example, is really difficult with traditional IR, not an issue in Raman. Glass, generally okay in Raman, of course, except for 785. And the spatial resolution independent issue, again, into Raman, it's completely independent. With IR, you have a strong dependency on wavelength. So if you look at that, it's no surprise that most labs will have two instruments, depending on the experiment, depending on the needs of the experiment. You may go to one or the other. In fact, you may even take the same sample and measure them on both, and then try with difficulty to find the exact same spot. But what we're doing with the O-PTIR technique is we're taking literally the best of those and putting them into a single microscope. Right, so that's my favorite animation, because it really does show how we combine best of infrared and Raman into a single platform. Okay. Over the last few months, there is some exciting developments, especially on the QCL front, because QCLs are an emerging and ever expanding and growing technology. So on the QCL side, the standard QCL that we offer is a 3-chip device from 1800 to 950, so covering the bulk of the infrared. If you're interested in the high wave number range, the OH-CH-NH region, there's a separate additional OPO laser, pump laser. But there are some options for the 4th empty slot of a QCL with some of these more custom options. And my personal favorite is the CH region chip, the CH chip, which covers the key CH region. So with doing that can be your 4th chip. So your one QCL will then cover your CH region and your fingerprint region, and that's a really exciting development. For those interested in the silent region, we've got some interesting bands there, the CD, triple bonds, nitriles, azides, azos. You've got the silent region combination, or if you want access to the longer wavelengths, go down as low as possible, down to about 800, you've got the wide range QCL. On the probe, which also doubles as the Raman excitation laser, we've got the 532, which is the standard, and the 785 is an option for those that are perhaps orange or reddish color, perhaps that are burn easily with the green. And just to give you some examples of how the spectra look compared to FTIR transmission reference libraries, I'm going to show you in a second FTIR transmission data that have been collected in transmission mode of thin films. And I'm going to compare that with O-PTIR data that have been collected in reflection of thick blocks. Right, and these spectra have been collected in about 5 seconds with about 0.5 micron, so 500 nanometer spot sizes. Right, so that's polypropylene. So in red, we have the FTIR reference library spectra, and in black, we have the spectra collected with O-PTIR in reflection mode of a thick block. Right, so pretty good match. And the same goes for PE. Polyethylene, the same goes for PET, nylon, and polystyrene. So it's pretty clear that the O-PTIR spectra, whether they're measured in transmission or reflection mode, reflection's easiest, are completely comparable to FTIR transmission libraries. Okay. We'll talk a lot about spatial resolution and the breakthroughs that O-PTIR brings there, and I'll remind everybody that spatial resolution can be approximated with the Rayleigh criterion, and it's proportional to the wavelength over numerical aperture of the objective. So for typical FTIR microscopes that have fairly long wavelengths, especially if you're working in the fingerprint region, the wavelengths are quite long. The numerical apertures are moderate to poor sometimes, so that means, say for the best FTIR microscope out there at the moment, at 1,000 wave numbers, you're looking at around 12 microns of spatial resolution. Yet with the O-PTIR, because we have a very short wavelength probe and a high NA, the theoretical resolution is about 416 nanometers. And importantly, it's flat. Whether you're in the fingerprint region or in the high wave number region, it's the same. And this equates to about a 30x improvement if you're looking at something at, say, 1,000 wave numbers. Okay, but perhaps the most exciting thing that we're bringing to the industry, into the field, to the users, is the fact that we can now do simultaneous infrared and Raman. And this is an absolute world first. It's incredibly exciting. So that really means same spot, same resolution, same sub-micron resolution at the same time from a single measurement. So if I were to take you through the schematic here, we've got the infrared tunable laser Your QCL typically, that is shone through and focused through this Cassegrain style objective, a reflective objective. We bring at the same time the green light, the probe beam, that's also collinearly focused onto the sample. That's where your photothermal magic happens. The reflected light is collected by the same objective and goes onto a visible detector for infrared spectral signals extraction. But any of the Raman photons that have been generated, remember we're using a Raman grade probe beam here, so there's Raman scattering whether we like it or not, whether we want to use it or not, it's happening. So we use a dichroic filter to pick off only the Raman scattered photons and send them off to a Raman spectrometer from where we collect the Raman spectra, and all of the other photons go on for visible detection for the infrared spectral extraction. So it's actually an incredibly efficient means of collecting Raman and collecting infrared without compromising either of the two measurements. And this takes full advantage of the complementarity that IR and Raman brings. It's confirmatory as well, so the infrared confirms the Raman and vice versa. It's a single instrument. It just gives you more thorough sample characterization. Okay. So with that background in mind, I'm going to switch now to giving you a quick overview of some applications. But before I do that, I just want to highlight the publications are on the rise. So this is incredibly exciting for us. We've gone from 1 or 2 publications in 2018 and 19. We had about 14 last year. We're into the low 20s this year, and by the end of the year, I'm expecting over 30. So that's a positive sign for the adoption of the technique and validation as well of the technique. And perhaps our biggest paper so far has been one from Oxana Klementieva, where she used the O-PTIR technique to highlight some polymorphic forms of amyloid aggregates directly in neurons within the context of Alzheimer's disease research. And this is perhaps my favorite figure from that paper, and the fact that we can differentiate at around 282 nanometers 2 spectra, one that shows a lot more beta structure to the protein. Another one from early last year, I think it was looking at some biodegradable polymers, some laminates really, of PHA on PLA, where they were interested in the interface. So we did a line scan here at 100 nanometer step size, 100 nanometer step size, let's repeat that. And it's in a single measurement generating O-PTIR and infrared spectra and Raman spectra. If we look at the infrared in a bit more detail, we can see that each one of these spectra are different and separated spatially by 100 nanometers. In this case, because we know the peaks of interest here, 1725 and 1760, we can do single frequency imaging and only image for those peaks of interest. Okay, moving on quickly, fibers. Anyone who's done fibers with infrared will know, certainly in reflection, that it's impossible. It just doesn't work. But whether we're working with 20 micron fibers here or 800 nanometer fibers, the spectra are diameter independent with excellent signal noise, no artifacts, and this is the raw spectra you get out of it. Okay. Very quickly, some phase dispersions on polymers. Initially starting with a fairly coarse image, and it's funny how nowadays I think of 500 nanometer step sizes to be fairly coarse. Doing a ratio between two peaks. If I go in there and do that at 100 nanometer step size, which is generally how I like to operate, the level of detail is exquisite. Some of these smaller dots here are sort of 200-300 nanometers in size. And if we look at spectra on and off these hotspots, you can clearly see the origin of the contrast in these images. Live cell imaging presents itself as a huge opportunity with this sort of technique, because it works so well in water, unlike regular IR. So this was a cheek cell experiment where I simply scraped my inside of my cheek with a toothpick, put that onto a calcium fluoride slide, drop of regular water, a calcium fluoride cover slip on top, and then put that into the microscope and collected some random spectra. And the spectra look like this. That's exactly how they come out. And notice that there were some peaks of interest, a lipid peak, of course there's protein everywhere, and some nucleic acids. So when I do a 3-frequency image, so 1085, 1551, and 1740, and then color them in an RGB, this is what you get. It almost looks like a fluorescence image. So you've got nucleic acids, not surprisingly, in the nucleus, and we can see lots of lipid inclusions in the cytoplasm. This was collected actually at 500 nanometers. step sizes. If I were to do this again, I'd do that at 100 nanometers. Okay. And this works well because O-PTIR has a low water background. That's because water actually has a high heat capacity and therefore has a low temperature change. And that makes it less visible than the other materials. Okay. Some breast tissue examples. Here, this was a case from the guys at Exeter Uni, where they came to me saying, here's a sample. I've measured this with FTIR imaging before. We think there's we can see some broad areas of calcifications. And my initial single point measurement in this area did not show any calcification. So I thought, all right, let's do something different. Let's start by imaging a single frequency at the calcification peak. And when we did that, it was a wow moment because unbeknownst to them and to anyone else, it seems as though these calcifications were really small, sort of 5 microns average size, even less. And of course, with an FTIR instrument at 1050, or even a regular QCL type of instrument at 1050, your spatial resolution is going to be on the order of 10 microns. So if your instrument resolution is 10 microns there, you certainly won't be seeing any of these features. It will look like a blur. But with the sub-micron resolution of O-PTIR, we were able to resolve all of these features very, very clearly. And then when you go in there and target some of these points and the points in between, you can see again the origin of the chemical contrast. And the spectra, of course, become a lot more pure, quote unquote, because there's without any of that spatial blur. Okay. Microplastics, that's an area of ideal fit, and there's some early work out there in the literature there as well. This is one of my favorite internal sort of model experiments we did, where we looked at different polystyrene beads ranging from 900 nanometers up to about 10 microns, and also a PMMA bead of 3 microns, doing this in salt water as well. So if you dry that onto a calcium fluoride substrate, you'll get salt crystals, you'll get all of the beads mixed together, and if anyone's tried measuring infrared in the presence of salt crystals, you know that salt crystals will completely ruin your signal. But with O-PTIR, you only get a signal if there's an absorbance. And since salt doesn't absorb, it's effectively invisible as far as O-PTIR is concerned. So the spectra in the bottom half here are all the polystyrene. You can see the spectra look like polystyrene, regardless of particle shape or size. So the spectra are consistent, as I've said there, regardless of particle shape or size. And in this case, being a simple binary mixture, taking those two frequencies, doing single frequency imaging, only those particles light up, not the salt crystals, not anything else. Okay. And with the help of Wiley and their know-it-all spectral searching engines and databases, with the simultaneous infrared Raman collection, you can also do simultaneous searching, which means at the same time you can search your Raman and infrared databases and have the results presented not as two separate lists, but as a 2D scatter plot, where the upper right-hand quadrant shows a data point or a hit that has a high IR hit quality index and also a high Raman hit quality index, so giving you confidence that that indeed is a good fit, one that you can trust. Here's an example of some microplastics that have been measured directly off a gold-coated polycarbonate filter. It's a very common way of filtering these, and whether it's a microplastic or whether it's some other particular from another application, the infrared and Raman spectra are measured simultaneously, of course, and then measure and search simultaneously. In this case, it's polycarbonate, so quite good matches in both IR and Raman. Over here, I've got an example from Kathy Goff, where she used the O-PTIR technique and took advantage of the IR polarization to examine collagen and collagen fibrils. Just very quickly, I'll point out that these fibrils are about half a micron, so we can image those, collect spectra of these fibrils, change the infrared polarization, perpendicular and parallel, and look at how the amide-1 and amide-2 bands change. And even some of the tissues we looked at are on calcium fluoride and on glass. It's something I'm quite excited about. With tissues in particular, glass becomes a viable substrate where none of the infrared radiation is obscured, unlike regular FTIR systems. Another glass example here, now we've got single cells. So the spectra here are single scans. So these are about 1 second's worth of measurements from about a half a micron spot on glass. right? So it shows again how glass doesn't hinder or block the infrared radiation. Sub-micron features are easily detected, and this was also done with this new laser, so kind of showing off the CH reading here as well. So in fact, I took advantage of that by looking at lipid chain length, taking an image of the CH2 to CH3 ratios at 100 nanometer step sizes, and you can see some pretty strong contrast indicating that there are indeed differences in the lipid chain lengths and also lipid to protein ratios. Perhaps we're seeing some nuclear membranes there. Not too sure. Okay. Peter Gardner's group at Manchester University also recently published some live cells and also some fixed cells using infrared with concomitant Raman spectroscopy. So with regular FTIR, these are really difficult measurements. Typically, it work with limited path lengths of around 7 microns. But as I said before and demonstrated here, O-PTIR shows very, very low water background. So it's actually an ideal tool to do live cell imaging, just like Raman is as well. And they took advantage of this upside-down sandwich approach, I call it, where the cell is actually grown onto a calcium fluoride substrate. It is merged with a lower substrate. And that lower one, in fact, can be glass or calcium fluoride. Actually, it doesn't matter as long as it's visibly transparent with a drop of water in between. And here the path length doesn't matter because the infrared beam interacts with the cell first before it's in contact with the water, so there's limited attenuation. That's where the photothermal effects happen, and they're picked up by the probe beam, which is now measured in transmission mode with a detector on the underside. So on the left-hand side here, we've got some fixed cell measurements or spectra from the paper, and on the right-hand side, we've got some live cell measurements, again, of IR and Raman. Roy Goodacre from the University of Liverpool has also published recently looking at isotopically labeled bacteria, E. coli in this case, where they were fed different substrates some of them were labeled with carbon-13, some of them labeled with nitrogen-15, and these, when incorporated into the cell, these have profound impacts on the proteins in particular. So they shift. So you can see some of the shifts from amyloid-1 to amyloid-2, sort of in the order of 30, 40 wave numbers. And all of this information is used to extract out more information about the metabolism of these cells. But this is a pretty stunning result because this is the first time anyone's really shown any single-cell bacteria with infrared. There's some examples of Raman out there, but Raman, of course, isn't as sensitive to protein as infrared is. So now we're taking advantage of the sub-micron resolution together with the protein sensitivity of infrared to start enabling these sorts of measurements. Now, this is something I did on my own, but with Roy's sample. It's in fact showing off our silent region chip. So these are some deuterium-labeled E. coli cells. You can actually see the CD absorbances coming through there, and this is a single bacterium that we've imaged at 50 nanometer step sizes, and then they've taken spectra along the length. So we're now actually doing intracellular single-cell bacteria measurements, which I think is pretty amazing given that this is still in the infrared. And lastly, I'm going to show you here a simultaneous IR and Raman spectrum of, again, a single E. coli. These spectra are collected in about 20-second accumulations. So you can see already that how IR is a lot more sensitive. It's much better as SNR compared to Raman. But you can take advantage of the complementarity still by doing these simultaneous measurements. Okay, so my key takeaway is that with O-PTIR, we take infrared spectroscopy beyond the accepted limitations of the technique, and we bring the best of infrared and Raman into a single platform. With sub-micron, you see a lot more detail. It's non-contact, so there's no cross-contamination, there's no dispersive and scatter artifacts, little to no sample prep. New Q-cert ranges are expanding new spectral regions and opening up new measurement capabilities. And I'm just going to throw this out there. With the addition of wide-field epifluorescence modules, we can also now do co-located or fluorescence-guided O-PTIR. So we can actually do measurements on samples that have already been labeled fluorescently for better targeting of areas of interest. And I'm going to finish off with this, again, reminding you that IR+Raman is same spot, same time, same resolution. Or let me just change something. Thank you. That's the end of my part, and I'm now going to hand over to Oksana for the main part of this webinar. Thank you, Oksana.
Hi, thank you, Mustafa. Hi, everyone. Today is 18th of November, and I'm happy to talk about correlative submicron infrared and X-ray fluorescence used for molecular and elemental imaging of neurons. I am working at Lund University, and I am happy to present today. So the aim of my work to provide insights into correlative approaches that could be used to retrieve more information about sample. So here is an example. Neurons grown on the surface, and the same neuron was imaged using several imaging modalities. So we use optical, synchrotron X-ray fluorescence microscopy, optical photothermal infrared spectroscopy, and we combined all images and try to explain what happens in the neuron. So in this way, I believe that we could get much more complementary information about sample and explain better what can happen in the cell in a very complex environment. So a little bit about Alzheimer's disease. So I'm working in Alzheimer's disease area. So Alzheimer's disease is very common disease. Every 10th person develop Alzheimer's disease after 80 years. And unfortunately, this is very severe disease when patients lose neurons and lose memory. So the first case was described in 1906. And unfortunately, we spent a lot of time to understand how molecular mechanism of working, but unfortunately we still have no clue. Also, the first drug has been approved in this year. We still have a lot of questions how to prevent Alzheimer's disease and how to prolong healthy aging. So commonly to understand Alzheimer's disease, we use a lot of methods. Commonly used imaging techniques such as bright field microscopy, fluorescence microscopy, or MRI PET scan. Those imaging techniques allows to look where the protein located and to find amyloid proteins with the nanoscale resolution. However, none of those techniques allow to understand the structural properties of amyloid protein that are linked to Alzheimer's disease. So here you can see on this electron microscopy those amyloid fibrils. They are formed from amyloid proteins and they have very specific structure. Specifically, this is amyloid-beta sheet structures. And what is importantly and what is used in the present study, that those specific amyloid structures can be detected by infrared spectroscopy. Infrared spectroscopy is a very well-known technique. It exists many years, and a lot of research has been done using infrared spectroscopy. However, resolution of infrared spectroscopies is above micron. So for the current research, it means that I could get resolution from 5 to 10 micrometers. So, however, infrared spectroscopy was reborn for structural study in neurodegeneration disease by using a new technique that recently came to be used, such as optical photothermal infrared and also nano-infrared spectroscopy. So new technique allows to better detect amyloid proteins and their structure, allowing the resolution at sub-micron and nanometer resolution. So how it works? So we have still infrared we have still infrared that, that illuminates samples and cause cause thermal response. However, there are new methods of detection. One is optical photothermal infrared, when we use visible laser light to detect a response, sample response to the infrared light, and other technique that allows nanoscale resolution detection of infrared signal coming from the sample. So using infrared spectroscopy, in my group we did a lot of study, and we studied amyloid aggregation in tissue and in cells. So, and using infrared spectroscopy, we were able to show that amyloid protein aggregates, and what is important, that structural changes can be detected by infrared spectroscopy. So here we use normal infrared study normal infrared with a resolution that are about 5 to 10 microns. So what's specifically what we show that changes in tissue, in brain tissue, that can be detected by infrared precede changes that can be detected by other techniques. So it means that and this was very important achievement and very important discovery, that amyloid plaque in brain tissue, they do not appear like popcorn. They appear much before amyloid. Those changes can be detected, and that means that infrared spectroscopy is an important tool that can be used to understand molecular mechanisms that link to Alzheimer's disease. So also using infrared spectroscopy, we will study what happens in primary neurons that are deposited on calcium fluoride. And already with the resolution that is provided by infrared spectroscopy, we could observe that that changes in protein structure already happen at the neuronal level, much before the neuron died and amyloid plaque is formed. So, however, using infrared spectroscopy, it was not possible to study changes of amyloid structures directly in neuron. So for that application, we need resolution, special resolution that goes beyond micrometer, and that was possible using optical photothermal infrared spectroscopy. Using this new technique that appeared for several years, we were able to study amyloid aggregation directly in neurons. So here in this image, you could see location of the spectra, and that was used to detect amyloid-beta proteins that are misfolded already in primary neurons. So here you could see this is very interesting experiment. Here we decided to study how spectral intensity depends on the thickness of neuron. So this is cell body, the most thicker part of the neuron. And then we took spectra with the distance of 1 micrometer, and we could observe that the intensity of the signal is correlate with the thickness of the neuron, goes down with when neurite becomes thin. So it means that, so in that sense, we try to address the penetration depth of O-PTIR, and we showed that neuron is a sample that can be used for O-PTIR study, and intensity of spectra can be used to estimate the thickness of neuronal outgrowth. So However, what was interesting, we observed that O-PTIR is much less substrate sensitive, and measuring samples in reflection mode, we can use different sample substrate, and that came the idea to correlate O-PTIR measurements with X-ray fluorescence measurements. So X-ray fluorescence measurements were done at Synchrotron Soleil at the beamline nanoscopium, and this beamline allows to study elemental distribution in the sample. So beamline comes through this the setup of the beamline is following. The X-ray beam comes through the sample. However, detectors are deposited very, very close to the sample, approximately with 1 degree from the sample surface, and those two detectors, they collect fluorescence signal that comes from the elements. And scanning the sample, we could detect different elements in the sample. So what we did, we took neurons from the mouse embryo when the brain tissue is homogenized and neuronal suspension is seeded on the sample substrate. So those neurons are allowed to grow for 19 days. So, and when neurons are have developed branches, like on this picture, you could see that neurons have maturated and they develop branches and connected very well. And we could also here see that on the glass, which is typically used to grow neurons for all experiments related for neuronal studies, we could see that same type of branching and connections neurons form on silicon nitride. And silicon nitride, this is the substrate that can be used for X-ray fluorescent imaging. And because optical photothermal infrared is less sensitive for substrate in reflection mode, neurons were able to also to examine using optical photothermal in spectroscopy. So this is the sample, how it was prepared. So this is sample frame, which in our case was 10 by 10 millimeters with the window, silicon nitride window of 5 by 6 millimeters. So on top of that silicon nitride, neurons were seeded and grown for 19 days. And here, this window was used for 2 experiments. So first, we did optical photothermal infrared experiment because optical photothermal infrared is doesn't destroy sample. And after that, we used X-ray fluorescence experiment to study elements in the sample. So this is an example how elements in neurons can be detected. So red dots is iron, and white is chlorine. So since neurons were grown in the medium which contains sodium chloride, we could see that chlorine is present in and make visible neurons. So we use this image to see where in neuron is, for example, iron or copper. So why we selected iron and copper? Those two elements are tightly linked to Alzheimer's disease. There is theory that suggests that iron and copper can participate in Fenton reaction, and that Fenton, and because of Fenton reaction, there are oxidative species that cause oxidative stress. And some of the theory that trying to explain Alzheimer's disease, they indicate that oxidative stress could could launch amyloid aggregation. So one of our questions was if we could see amyloid structures together with iron or with copper. So this is an example how different elements could be detected in primary neurons. So we could look on iron, on copper, on zinc, and depending on experimental setup, other elements can be also selected, like calcium or sulfur or phosphate. So now in our setup, we use two different microscopes, and it was important to be able to find the same neurons. To achieve that purpose, we marked one corner on the silicon window, and then scan silicon window with the microscope, and we selected the neurons that we are going to use for the X-ray fluorescence and optical infrared. So, and we recorded the coordinates of those neurons. So as an example shown here, so we mapped that. So we mapped these neurons, and then we could find the same corner and using two microscopes and using the coordinates, we could go to the target. So, and then what we did, we imaged neurons using optical photothermal infrared. And using optical photothermal infrared, we recorded several maps at different frequencies. For example, 1656 to get total protein contribution. Then we recorded maps at 1628 to record the to locate presence of beta sheet structures. Also, we recorded maps at 7040. This band is known for the lipid oxidation that linked to Alzheimer's disease. So and after that, when all maps were recorded, we analyzed the sample, and for example, taking the ratio, we looked where amyloid-beta structures located and also where oxidized lipids are located. So after that, we found the same neuron using X-ray fluorescence and scan the same neuron using X-ray fluorescence. So here is an example of distribution of iron and copper. And what we could see, what could we observe? We could observe those big clusters of iron, while copper is distributed without clustering. So first, using previous experiments, we compared wild-type neurons and neurons that were used for Alzheimer's disease. And what was interesting, we observed that only in neurons that are models of Alzheimer's disease, we could observe that clustering of iron. However, we cannot see any change, any difference in copper. So, and now we wanted to look if those clusters somehow co-localized with amyloid-beta sheet structures. And using image overlay, so using masking procedure, we looked if those clusters co-localize with beta sheet structures and if those clusters co-localize with oxidized lipids. And actually, what could we see? We could see that iron, but not copper, overlays with beta sheet structures and with lipids. However, we could not see that all beta sheet structures contain iron and that all oxidized lipids are co-localized with iron. So this is interesting phenomena that opens new possibilities for further research if there is a difference between iron clusters that co-localize with beta sheet structures, and if there is a structural difference between those beta sheet structures that are co-localized with iron. So, however, in our study, we have not addressed that questions. So what we did, we showed the possibility. This was the proof of concept study if two techniques can be combined for biological research. So, and in summary, I would love to say that in our work, we demonstrated that optical photothermal infrared spectroscopy can be used for X-ray with X-ray fluorescence microscopy for label-free optical imaging to image amyloid beta sheet structures and metal distributions directly in cell at submicron resolution without chemical processing. So this was very important to preserve all metals and to preserve all amyloid structures without altering them using procedures that are needed for labeling of amyloid structures, for example. So with that, we using O-PTIR combined with X-ray fluorescence microscopy, we showed that amyloid beta proteins can form beta sheet structures in cultured neurons, and iron, but not copper, clusters co-localize with elevated amyloid beta sheet structures and oxidized lipids. So with that, I would like to thank you all for listening my lecture, and I hope with that example I demonstrated that more information can be retrieved from the sample and can help to understand molecular mechanism in a complex sample. So I would love to thank also Synchrotron, where the experiments were done, and I would like to especially thank you Photothermal Corporation for their help in learning techniques and understanding results, and also funds that allow the research. Thank you.
Thank you, Oksana, for that enlightening webinar and presentation. So we do have some time for questions, and I can see a few questions coming in. I think some of them will be for you and some for me. The first one looks like it's definitely for you, and that says, can you be sure that that the iron and amyloid are truly localized at the same three-dimensional point?
So yeah, this is very good question, and it actually shows that we need more, that it could be more complex than it could be more complex. So yes, indeed, IR penetrate the sample and X-ray penetrate the sample. And it could be that we have co-localization, spectral co-localization, but studied materials could be separated in the cells and not co-localize with that. So this is the possibility. However, it is known that amyloid protein binds iron, and that it is most likely that those iron clusters are co-localized with amyloid beta sheet structures. Also, analyzing size of these clusters and size of amyloid amyloid detected in cells, it is most likely that the size coincides and it's and iron co-localize with amyloid beta. However, I agree that more experiments are needed to truly demonstrate that that iron is is co-localized with amyloid-beta. We could, for example, try to isolate amyloid proteins and do elemental analysis of amyloid-beta isolates. However, the samples will be processed, and we could lose structural information, and we could lose ion metals. But yes, so I believe that amyloid-beta co-localize with iron, because amyloid-beta is metal-binding protein. So, however, it's this experiment does not show clearly co-localization of those two elements.
Okay, thank you for that thorough answer. I'm going to move on to the next question. This one is probably for you as well. Any damage from the lasers used, and how would you control that?
So it is possible to damage biological sample using using the laser, of course, and also using the X-ray beam. So this is definitely the possibility. Therefore, we used experimental setup when we first use infrared spectroscopy, and then we use X-ray spectroscopy. However, if we use high power of laser, we could also burn the sample. Therefore, in our experiments, we used extremely low energy of probe laser from 0.5% till 4%. So since measurements are so takes very little time, we have not observed spectral differences when we scan the same sample several times. So, but it is the possibility to damage sample with the laser if the laser set to high power. Therefore, it has to be studied before the experiments, the sensitivity of sample.
Thank you. The next one might be one for me. How does O-PTIR not suffer from fluorescence if you use a Raman laser? And that really goes back to my point earlier about how we use a Raman grade laser. Now that's pretty simple. The QCL infrared laser that we use is modulated. So that means that the signal that we extract out is also modulated. And that's separate from the Raman signal and also the fluorescence signals, which are not modulated. So by using a lock-in amplifier, we can efficiently extract out only the infrared portion that contains that infrared spectrum information, whilst completely excluding any fluorescence background signals. Then this probably will have to be our last question. What is the depth of sampling? And this is a common question, and one that I always think I should have mentioned during my presentation. And that, like many things in life, it depends. But as a rule of thumb, and it depends on the materials that are being measured at that point in time, but as a rule of thumb, as just a general rule of thumb, I'd say probably a few microns would be your depth of sampling, or depth of penetration. And with that, I'm conscious of time, that we have to go. And once again, Oksana, thank you for taking the time, I know, out of your busy schedule to give this informative webinar. So again, thank you. And thank you for everybody who's dialed in, and there's quite a few of you there, so yeah, really appreciate that. It's really encouraging to see. This will be posted on our website within the next few days for on-demand viewing, so you'll be able to pass it on to any friends and colleagues who you think might be interested. So yes, once again, thank you everybody, and hope to see you with our next webinar. Bye-bye.